Assistant Professor, Amity Institute of Organic Agriculture. Welcome and extend a very warm welcome to all the distinguished dignitaries, invited experts, faculty members, research scholars, and participants joining us today for the 5-day faculty development program on climate resilient agriculture and allied technologies for net zero and socially inclusive framework organized by the Amity Institute of Organic Agriculture, Amity University, Noida, Uttar Pradesh. I would like to extend a heartfelt thanks to our Honorable Founder President Dr. Ashok K. Chauhan sir, Honorable Chancellor Dr. Atul Chauhan sir, Honorable Vice Chancellor Professor Dr. Balwinder Shukla ma'am, the Director General of the Amity Food and Agriculture Foundation Dr. Nutan Kaushik ma'am, Director of Amity Institute of Organic Agriculture Dr. Doili Sanyal ma'am, and all the eminent speakers and guests from all over the world who have joined us today for their unwavering support, guidance, and blessings. It gives me immense pleasure to begin this academic gathering aimed at capacity building, knowledge exchange, and advancing excellence in sustainable agricultural practices and research. This faculty development program has been designed to enhance participant understanding of emerging trends, innovative technologies, and interdisciplinary approaches essential for strengthening agricultural teaching, research, and extension. The key focus of this FDP include climate resilient agriculture frameworks and technologies, soil, crop and water management for climate adaptation, role of bioresources, bioinputs and organic farming, AI, IoT, big data and satellite tools, and policy and community models. Through these all technical sessions, expert lectures, panel discussions, and interactive modules, the FDP aims to build strong conceptual foundations while
Deepshika, you are muted. Deepshika, you are muted. Unmute yourself, please. Dr. Deepshika, unmute yourself.
Yes, sir. So can you please let me know where did where the thread was broken? Was I mute from the start?
No. You
Okay.
had spoken about the themes. Now you can proceed.
And I would again like to repeat the key focus areas of the FDP. They are climate resilient agriculture, CRA frameworks and technologies, soil and crop and water management for climate adaptation, role of bioresources, bioinputs and organic farming, AI, IoT, big data and satellite tools, policy and community models. Through the technical sessions, expert lectures, panel discussions, and interactive modules, the FDP aims to build strong conceptual foundations while promoting practical, field-oriented knowledge. To formally commence today's proceedings, I am pleased to invite Dr. Doili Sanyal, Director, Amity Institute of Organic Agriculture and Chairperson of this FDP, to kindly address the gathering and highlight the importance of this faculty development program in strengthening academic excellence and promoting sustainable agricultural innovations. Her leadership and vision have been central in conceptualizing and organizing this program. Ma'am, we request you to please share your valuable insights. Welcome.
Thank you, Deepshika. Am I audible?
Yes, ma'am.
Yeah. A very good morning to everyone. I extend a very warm welcome to our distinguished speakers, academicians, researchers, faculty members, scholars, industry professionals, and participants who have joined us from different institutions and organizations. It is truly encouraging to see such an overwhelming response this FDP has received, where enthusiastic minds committed to the future of agriculture and sustainability have gathered to deliberate on this important topic. I'm delighted to share that around 250 participants have joined this FDP, representing an exceptionally diverse academic and professional community from north to south of India. Participants have connected with us from nearly 190 institutions, industries, and organizations across India, including central and state universities, private universities, ICAR institutes, research organizations, industry representatives, and independent scholars. What is particularly encouraging is the diversity of the participants' profile. For us at Amity, Institute of Organic Agriculture, this enthusiastic response sends a strong message. There is a growing recognition that challenges posed by climate change can only be addressed through collaboration across disciplines, institutions, and sectors. Climate resilient agriculture is not merely an agricultural issue nowadays. It encompasses environmental sustainability, technological innovation, policy development, social inclusion, rural livelihoods, and food security. What excites me most is that over the next 5 days, this program will bring together experts and learners from different backgrounds, creating a unique platform for exchanging ideas, experiences, and innovative solutions. Some of the collaborations and discussions that begin during this FDP may well evolve into a future research project, institutional partnerships, impactful interventions for sustainable agriculture. As someone deeply involved in organic agriculture, sustainable farming systems, and climate resilient development, I firmly believe that the future belongs to integrated approaches that combine scientific innovations, traditional knowledge, ecological sustainability, and social inclusiveness. This FDP has been designed precisely with that vision in mind. As we begin this program today, I would like to take a moment to reflect on why the theme we have chosen is so important. Climate change is no longer a challenge of the future. It is something we are experiencing every day. Unpredictable rainfall, rising temperature, extreme weather events, declining soil health, increasing pressure on natural resources are directly affecting the agricultural system and farming communities. As educators, researchers, and professionals, we have a shared responsibility to develop solutions that not only increase productivity, but also build resilience and sustainability. Agriculture today must do much more than produce food. It must conserve natural resources, reduce greenhouse gas emissions, enhance biodiversity, improve livelihoods, and contribute towards achieving national and global climate goals. This is where the concept such as climate resilient agriculture, net zero pathways, precision technologies, artificial intelligence, genomics, bioinputs, and socially inclusive development frameworks become extremely important and relevant. One of the reasons I'm particularly excited about this FDP is its multidisciplinary nature. Over the next 5 days, we will have the opportunity to learn from eminent experts from India and abroad who have made significant contributions in their respective fields. The program brings together perspective from plant breeding, genomics, digital agriculture, climate-smart technologies, soil and water management, microbial solutions, policy frameworks, and sustainable development. Such interactions are invaluable because they allow us to move beyond disciplinary boundaries and look at agriculture challenges through a broader lens. For me, one of the most important aspects of any FDP is not only what we learn from the speakers, but also what we learn from one another. Every participant brings unique experience, ideas, and insights. I therefore encourage all of you to actively engage in discussions ask questions, share your perspectives, and use this platform to build meaningful academic and professional networks. I firmly believe that the future of agriculture will depend on our ability to integrate science, technology, traditional wisdom, and social inclusiveness. Climate resilience cannot be achieved through technology alone. It must be involving the local farmers, rural communities, women, self-help groups, youth, policymakers, researchers, and industry stakeholders working together towards a common goal. On behalf of Amity Institute of Organic Agriculture, I would like to express my sincere gratitude to all our distinguished speakers for graciously accepting our invitation and sharing their expertise with us. I also thank the organizing committee, faculty members, and staff whose hard work and dedication have made this program a possibility. I hope that the next 5 days will be intellectually stimulating, professionally enriching, and personally rewarding to all of us. May this program inspire new ideas, collaborations, research initiatives, and practical solutions that contribute towards climate resilient, sustainable, and inclusive agricultural systems. Once again, I warmly welcome each one of you and wish you a fruitful and enjoyable learning experience. Thank you so much. I wish the program great success. Over to you, Deepshika.
Thank you so much, ma'am, for your enlightening words. Now I take the privilege of inviting Dr. Nutan Kaushik, Director General, Amity Food and Agriculture Foundation, a distinguished leader in agricultural research and innovation, to share her remarks and guide us with her vision for the future of sustainable agriculture. Ma'am, we warmly welcome you to address the audience, please. In the meantime, can I please request all the participants to turn on the cameras?
Sorry, I was mute actually.
Okay. We can. Welcome, ma'am. Welcome, ma'am.
Thank you, Dr. Deepshika and Dr. Doyle and DK for this invite. A very good morning to all of you, to distinguished guests, eminent speakers, respected faculty members, researchers, scholars, industry professionals, and joining us from across India and abroad. So it gives me immense pleasure to extend a warm and heartfelt welcome to all of you to the 5-day faculty development program on climate resilient agriculture and allied technologies for net zero and socially inclusive framework, organized by Amity Institute of Organic Agriculture under the aegis of Amity Agriculture and Allied Domains of Amity University, Uttar Pradesh, Noida. At the outset, I would like to express my sincere gratitude to Honorable Founder President, Dr. Ashok K. Chauhan, for his visionary leadership and unwavering commitment towards excellence in education, research, innovation, and sustainable development. His vision continues to inspire us to address global challenges through interdisciplinary knowledge and impactful action. To the benefit of the audience, I would like to acknowledge here that agriculture is very close to our Honorable Founder President, sir. And it is because of his vision only that he created the agriculture discipline right from the inception of this university. Amity is the only private university who has the agriculture, not only one institute, but we had right from the beginning around 10, 12 research institutes and centers dedicatedly working in the area for agriculture and allied sciences. And we have 12 research centers and institutes who are working in this area. And today we gather at a crucial juncture in human history, where climate change, resource degradation, biodiversity loss, and increasing pressure on food system have made it imperative for us to rethink the way agriculture is practiced, taught, and researched. Agriculture must not contribute to environmental sustainability, carbon neutrality, and social inclusiveness. As highlighted in the program overview, climate resilient agriculture is emerging as a key strategy to address greenhouse gas emission, climate uncertainties, and the need for the sustainable livelihood. This FDP has been carefully designed to provide participants with comprehensive insights into climate resilient agriculture through global perspective, advanced technology, policy frameworks, and practical approaches. Over the next 5 days, participants will engage with topics ranging from climate smart soil, crop water, post-harvest management, precision agriculture, artificial intelligence, genomics, genome engineering, and climate smart breeding and strategies. We are privileged to have us with an outstanding lineup of the eminent national and international experts representing renowned institutions such as ICR, IRI, NBPGR, NIPGR, BRIC, NABI, CIMIT, IRI, INA, Murdoch University, BHU, and so on. They are all leading research organizations from where we have picked up the speaker, the organizers have picked up the speakers. Their expertise and experience will undoubtedly enrich our understanding and inspire new ideas for research, teaching, and innovation. This program aligns closely with India's vision of Viksit Bharat 2047, emphasizing sustainable agriculture development, net zero pathways, climate resilience, technological innovation, and inclusive growth. It also reflects our collective responsibility to empower farmers, strengthen food system, and build resilient agriculture communities capable of meeting the future challenges. I would like to acknowledge the dedicated efforts of organizing committee under the leadership of Dr. Doily Sanyal, Director AIUA, Dr. D.K. Chaudhary, convener, and the team of co-conveners and faculty members whose meticulous planning has made this program possible. I'm delighted to see enthusiastic participants from faculty members, scientists, researchers, scholars, government officials, NGO representatives, and industry professionals. Your presence reflects a shared commitment to advancing sustainable and climate resilient agriculture system. As we commence the FDP, I encourage all the participants to actively engage in the discussions, interact with the experts, share experiences, and explore opportunities of collaboration. The true success of a program lies not only in acquiring knowledge, but also in transforming that knowledge meaningfully into actions that benefit agriculture, society, and the environment. Once again, I extend a very warm welcome on behalf of our Honorable Founder President, Sir, Dr. Ashok Chauhan, our Chancellor, Dr. Atul Chauhan, Sir, our Vice Chancellor, Mentor, Dr. Professor Balwinder Shukla, and on my personal behalf and on behalf of all the faculties and students and scholars of the FDP University, I wish to extend a great welcome to all of you. I also welcome distinguished speakers and the participants to this program. I wish the program a great success and hope that the next 5 days will be intellectually stimulating, professionally enriching, and highly productive for everyone involved. Thank you very much. Jai Hind.
Thank you so much, ma'am. Thank you for your remarks on this FDP. Now, next I invite Professor Dr. D.K. Chaudhary, convener of this FDP, to apprise the participants with overall guidelines, instructions, and regulations for participants. Sir, I request you to kindly brief the participants about the FDP structure, rules, and expectations.
I think
Over
few
to you,
minutes
sir.
that do I have. Thank you, Deepshika. At 11 sharp, we shall move ahead. Before this, I would say that the topic conceived by us, by the Agri staff, Agri team, the climate resilient agriculture and allied technologies for net zero and socially inclusive framework. We were thinking a few important points relate with this. How do microbes do in the soil and what are their peculiar characteristics that soil makes worthy? How to use soil microorganisms to enhance the productivity and ameliorate the habitat imposed stresses whereby crops grow well under the climate resilient agriculture theme. By keeping this view in mind, we all sat together and decided to move ahead and invited the eminent speakers who are devoting their research in the particular themes and we split the entire thematic area into the sub-themes, the 5 sub-themes I think Deepshikha has already defined altogether. Alongside, much has been discussed by Dr. Doily and Dr. Nutan Kaushik, Madam. What left for me? I would say that I shall introduce speakers, what kind of speakers that we invited to unravel the perplexity of the climate resilient agriculture. On this day, that is a June 1, we have Dr. Enrico with us from Italy, Dr. Berenci from BHU, Dr. BP Mallikarjun Swamy from Philippines, Dr. Patricia Galaffi again from Italy, and they will enlighten us on the topic climate resilient agriculture concepts, framework, and the global perspectives. Besides this, the second day will be devoted to the exploration of the microorganisms, what do they do, and how diverse they are in the soil, whereby they, enhance the soil productivity alongside the crop productivity as well under the biotic and abiotic stresses. For this, the Dr. H.P. Singh, we invited him, and he is truly involved in this area for last 50 years. And beside that, Dr. Siddharth Tiwari, and certainly he shall provide glimpses on the CRISPR technology because he is working on the CRISPR aspects, belongs to the BRIC NABI. Dr. Devbandhu Padhyay, Mahapatra, she is working on the post-harvest engineering. She will provide some glimpses related with this aspects. Besides that, and all together, we have Dr. Arun Joshi, and he did work at CMIT and the BISA as well, and where he will enlighten us about the agricultural extension aspects. Dr. Bhupinder Giri, he will provide insights on the mycorrhizal aspects. What do mycorrhizal do in the rhizosphere, whereby the plants may ameliorate under the habitat-imposed stresses. AI deployment nowadays, the peoples all around globes are deploying the AI in curricula as well. So we provided some glimpses on the AI technologies in agriculture. So Dr. Shankar Goenka will provide glimpses on the digital agriculture and climate smart decision supports. Beside that, we shall move ahead with the gene editing aspects, whereon the glimpses shall be given on us by the Dr. Niti Sanand Mishra ji from ICJB, New Delhi itself. Alongside, we shall move ahead, just like progressions towards the deployment of the AI in agriculture. A few brilliant faculty that do resides in Amity itself, Dr. Akash and Dr. Monica, shall provide glimpses on the AI. Beside that, we shall move ahead by deploying the genomics and genome engineering aspects. For these sessions, we have Dr. Rupesh with us, and he is working at Central University of Haryana, India, and working on aspects on the genome approaches, climate resilient and nutritionally safe crops by deploying the genome engineering. Beside, we have Dr. Reyazul Rauf Mir from Australia. He will provide glimpses on enhancing nitrogen use efficiency. No doubt, very, very interesting topic, along with other eminent speakers. Dr. Dinesh Kumar from IRI New Delhi. How does organic farming play an important role to make a socially inclusive framework? That is, again, important aspects that will be unraveled. Alongside Dr. Saloni Mathur, break NIPGR, and she will provide glimpses on the interplay between the RNAs and stress responsive pathway for the climate resilience in plant. Certainly, the gene editing aspects playing important role nowadays, along with the, if I use one term, polyphasic approach, polyphasic taxonomy, right? Phenetic and the genetic aspects all together, we shall enhance in this FDP. Dr. Prashant Vikram, again, genome-driven climate smart agriculture. On the last day, we shall move ahead with the Dr. Value. And again, he is working at CMIT Mexico on the wheat breeding aspects. How do we can provide the food security globally by deploying such billions of populations under the habitat imposed as to what kind of the gene modification is taking place in the wheat and maize. So he will provide some glimpses. Alongside, we shall move to Dr. Navtesh Singh Bains, and he will provide the genetics and genomic strategies for developing climate smart crops. Last sessions will be delivered by Dr. Nagin, and he is from the Genomic Foundation, JC Bose National Fellow. He is from ICR NIPB New Delhi, and he will provide some glimpses on the gene sequencing for innovating nutritional and stress-related traits among to produce the climate smart crops. And on that day, we shall see a Q&A session also. Some assessment will be imposed to the participants, what kind of the learning practices they have learned, and thereupon we shall move to the valedictory session. So Dr. Deepshika, this is just from my side. I did not take much time and hope we are at scheduled. And Dr. Deepshika, it's up to you now. You can invite first speaker to deliver a talk. Thank you.
Thank you. Thank you so much, sir, for the detailed
Deepshika.
description of the FDP. Yes, ma'am.
Deepshika, please do inform the participants about the rules and regulation, percentage attendance, and where they will see the attendance and exam and all that, please, so that they do not falter and do not receive the certificate.
One more line I would like to describe. The e-certificates will be issued to participants if they appear more than 100 or more than 90% attendance, not 100. 100 is belong to the outstanding participants. So we appreciate the participants if they show appearance over here and show attendance more than 90%. So they will get certificate, e-certificate. And Q&A sessions we already defined on that day. And more than 60% satisfactory response must be given by them to us, so they will get certificate. Thank you.
Thank you so much, sir, for letting all the participants know that at the end of the FDP, there is going to be an exam, and 60% pass marks is there for being qualified for the e-certificate. So please make sure that all the participants, you attend the at least 90% attendance is there in all the sessions and all together. And and you attend the exam that is going to be there at the end of the FDP, and 60% is required for the e-certificate. Thank you so much, sir. And before we move ahead to the technical programs, I request all the participants to please turn on the camera so that we can take a group picture of everyone. Please turn on the cameras.
You can drop a message on chat box, Deepshika.
Dr. Tarun, I would request you to share the QR code for attendance in the chat box as well, other than WhatsApp group.
I will again announce and request all the participants to please turn on the cameras for a group photograph, please.
Good morning, Professor.
We can take one more minute to wait for all the participants to turn on the cameras. Many of the participants have already turned on the cameras, and I would also request the others to please turn on the cameras. Thank you so much. So with this, we will move ahead to the first technical session of the day based on the theme Climate Resilient Agriculture, Concepts, Framework, and Global Perspectives. Our first speaker, Dr. Enrico Porcedo, Emeritus Professor is a distinguished plant geneticist and is from University of Tucicia, Viterbo, Italy. He is a distinguished plant geneticist and globally acclaimed wheat scientist known for his pioneering work in durum wheat genetics, molecular breeding, and plant genetic resource conservation. A long-standing faculty member at the University of Tucicia, Italy, he has shaped the research in cereal genomics, quality traits, gluten proteins, test tolerance, and utilization of genetic diversity for sustainable agriculture over more than 5 decades. With over 200 influential publications, 6,000 plus citations, and an H index of above 40, Dr. Enrico's contribution to wheat storage protein characterization, molecular markers, QRT-PCR standardization, linkage mapping, and stress-responsive gene studies have become foundational in modern cereal biotechnology. He has also played a vital role in documenting and conserving wheat germplasm across the Mediterranean, Ethiopia, and West Asia, strengthening the global food and nutritional security efforts. Integrating classical genetics with cutting-edge molecular and genomic tools, his research has enabled the development of wheat cultivars with superior quality, resilience, and climate adaptability. He has been deeply involved in international collaborations with ICARDA, European wheat programs, and biodiversity initiatives, and continues to influence global research in precision breeding, genomic innovation, and climate resilient agriculture. It is now my privilege and pleasure to invite our distinguished speaker for the much awaited lecture. Dr. Enrico, sir, please.
Cos'è questa? No. Qual è?
We can see the presentation. Yes, sir.
Qual è Fabio? Qual è? Qual è adesso?
Hands off.
Schermi te. Ah, qui. Good morning,
Good
Mr.
morning,
Chair.
sir.
Good morning.
Good morning, Professor.
Let me begin by saying that I am deeply honored to take part in this important event dedicated to, as I understand, to the education and training of young people preparing to advance agriculture. Since the 1970s, I have had the privilege of visiting India several times, collaborating with scientists, supervising Indian PhD students, first at the University of Birmingham, UK, and more recently in Italy, including Dr. Kartikeyan. Mr. Chair, I'm about to undertake a difficult and delicate task, to speak about Indian matters before an Indian audience. I understand the value of hearing challenging ideas, but do not be surprised if you hear some rather far-fetched ones. Let me ponder some points. Over the past 6 years, Indian agriculture has achieved something extraordinary. Food production has increased 4.4-fold, while the population has grown by about 3.2 times, and this has been achieved without any significant expansion of agricultural land. Today, Indian agriculture provides around 1.5 gigatons of crops and livestock products every year. This stimulates transport, processing, distribution, and retail, and contributes to the creation of a vast food system that provides employment for about 450 million people, whose incomes support the livelihood of almost 900 million people. Despite these achievements, about 16% of the Indian population remains undernourished, and 45-50% consume diets that are unhealthy or nutritionally inadequate. But this situation is not due to the food system. In fact, over the past 6 decades, Indian food crops production has increased substantially, stimulated by major improvements in crop yields. However, the larger gains have occurred in crops that mainly provide dietary energy, such as wheat, rice, maize, roots, and tubers, whereas pulses, one of the principal sources of plant proteins, have experienced much more limited growth. In addition, the cultivated area devoted to this crop, or this set of crops, has not been recovered from the reduction that occurred in favor of cereals, in spite of the stimulating governmental programs. The production of fruit and vegetables, which provide vitamins, minerals, bioactive compounds, antioxidants, and hydration, has increased mainly through the expansion of the cultivated land rather than through higher yields. At the same time, between 30 and 50% of agricultural land is estimated to be degraded, causing yield reductions of 15–25%. Extensive agricultural areas have already been abandoned. Thus, the environmental foundation of agriculture are under stress. Soil quality is deteriorating, biodiversity is declining, agriculture already uses 80% of national freshwater resources and agriculture contributes more than 20% of greenhouse gas emissions. And more importantly, agriculture faces an additional challenge, the climate change. Climate change is already affecting agricultural production. Higher temperatures, changes in rainfall patterns, and increased frequency of extreme weather events reduce crop yields. Scientific estimates suggest that wheat production in India could decline by more than 6% for every 1 degree centigrade increase in temperature. Climate change also causes rapid shifts in growing seasons and increases vulnerability to pests and diseases, as shown by the rapid spread of wheat stem rust, AUUP 99. Let us look at future. Looking at future, the challenges become even clearer. By 2050, India's population is expected to reach about 1.7 giga people, and more than half of them will live in cities. Food demand may increase by between 80 and 100%. Rising incomes and urbanization will change diets, increasing demand for more diversified and higher value foods. Meanwhile, competition for water will increase, production risks will rise, market access and affordability will change, access to land may become a serious problem. These pressures led to one clear conclusion: business as usual is not an option. Agriculture must undergo a profound transformation. And on this respect, I would like to draw your attention to 6 points. Human resources. Agriculture consists of many interconnected factors. Understanding those factors, their interconnections and their problems, scientists and technicians will be able to interact with the farmers and benefit from each other's knowledge, promoting a model of knowledge-intensive management that integrates traditional and emerging technology options, increase productivity, ensure production stability and safety, and preserve resources and ecosystem services. This transformation requires a new generation of biological, physical, and social scientists, as well as farmers prepared to govern change, identify difficulties and problems related not only to agriculture, but also to sociology and anthropology, and possess sufficient implementation capacity. The preparation of these human resources, I feel, represents a major challenge. The second point concerns the soil. The single most biodiverse habitat on Earth, a complex ecosystem regulating several unique functions essential for crop productivity, ecosystem stability, and human and planetary well-being, as had been recorded a few minutes ago. As a storage of carbon, soils help to regulate CO2 and other emissions. In India, 168 million hectares of arable land produce 86% of national food supply, but about 30% of the soils are degraded. This leads to production losses of 25% and decline in ecosystem services. Without change, by 2050, large areas of Indian soils, as well as terrestrial ecosystems and food production systems, will deteriorate further. This makes it essential to strengthen research on soil fertility, nutrient balance, and soil biodiversity, as well as to develop technologies for soil restoration programs. The third point concerns the water resources. All we know that water is basic ingredient of life and the most critical resource for both agriculture and human survival. Climate change will intensify the water cycle, increasing the severity of droughts in some regions and causing dramatic floods in others. Indian agriculture already utilizes 80% of freshwater resources. Farmers are increasingly relying on groundwater reserves. Indian area equipped for irrigation amounts to 75 million hectares, with an increase of 25-30% since the beginning of the century. But more than 60% of water catchments have reached a critical point or are endangered. By 2050, population growth and industrial development will intensify competition for water and make this resource even greater challenge for sustainable food production. New water management strategies must therefore be developed together with breeding crops with higher water use efficiency. The fourth point concerns crops and livestock protection. Meteorological events, pests, diseases compromise crops and livestock production. Statistics indicate that 25 million tons of produce are lost before harvest every year in India. Farmers attempt to control biological threats by using agrochemicals, but these generate environmental and health costs. Additional risks are associated with the microbial agents and their mycotoxins, which may occur in uncooked food, fruit, vegetables, etc., negatively affecting safety and contributing to chronic disease, cancers, and impaired child growth. Research programs should therefore aim to develop climate-resilient crops, pest and disease-resistant plants, greater crop diversification, including the species domestication or redomestication, and study of novel food options. The fifth point concerns innovation and technology choices. Research activities are continuously developing technologies that can contribute to the sustainable production of safe food, including perennial cereals, associations between cereals and nitrogen-fixing bacteria, remote sensing, nanoparticles for monitoring plant health, animal vaccines, and many others. Their rapid dissemination will allow agriculture to benefit from frontier areas of sciences as occurs in many other fields of human activities. That will also significantly enhance sustainable food production. But these advances must comply with clear safety instructions and international regulatory agreements. Some of these technologies are scientifically neutral, but not resource neutral, and their use may increase farm costs. In India, around 155 million small farmers cultivate less than 2 hectares and yet produce about 50% of the country's food. How can they benefit from these opportunities? That is a clear point. Research should explore options for devising appropriate policies that stimulate collective actions. Education and training aimed at improving technological competence, including awareness of environment and health risks, are equally important. Education and training on this point is extremely important and urgent. The sixth and final point concerns the socio-economic aspects. The food system is complex. Food may be produced, processed, and consumed in different parts of the country or even in different countries. India is still importing a certain amount of pulses. This trend has contributed to increasing food quality, quantity, and diversity, but it has also generated socioeconomic issues that deserve clear attention. Food markets, through advertising, information, and labeling, may influence consumer choices and thereby affect farmer decisions regarding which species to cultivate, which technologies and management practice to adopt, etc. Statistics indicate that Indian diet consists predominantly of starch, with significant deficiencies in compounds provided by pulses, vegetables, and fruit, all of which could contribute to a more balanced diet. Their cultivation could also stimulate soil fertility regeneration. Agriculture in India remains the sector with the highest level of informal employment. Innovation and increases in labor productivity might relocate millions of people to other sectors. It may be necessary to promote the integration of small farmers into value chains by improving access to markets, information, inputs, and developing downstream segments of the food system. This change may require coordinated strategies capable of ensuring coherence and maximizing synergies while avoiding the promotion of unsustainable agriculture and disappearance of small farmers. Research activities should provide information needed to support this strategy. We must recognize that the challenge is not only agricultural, it concerns the entire food system. And this brings me to a simple conclusion. The future of future research and education should not simply be to increase agricultural production. It should be to transform the entire food system, adopting ecological aspects, technical aspects, social economic realities. This should ensure food security environmental sustainability, and prosperity for present and future generations. Mr. Chairman, let me conclude by expressing my best wishes for students and professors active during the next years in this difficult job. Thank you.
Thank you so much, sir. Thank you for your knowledgeable insights and enriching lecture. We will now open the forum for any questions from the participants. Dear participants, if there are any questions, please type them in the chat box so that I can put forward to our respected speaker.
You can ask also. Deepshika,
Yes, sir.
very nice. Dr. Enrico, thank you, sir, for coming over here through Dr. Karthik. Thank you, sir. Am I audible to you, Dr. Enrico?
Yes?
Yeah. I was thinking, echo, I think echo is going on. Unmute yourself. Two things came in my mind. A Sustainable Development Goal 2 and 13. So
Could you speak louder?
Hello. Am I audible?
The echo from that side, sir, not here. I muted.
Could you speak louder?
Yeah. Dr. Enrico, am I audible to you?
Yes.
Yeah. Upon your presentations, what I got, we have to go for the agri transformation, very much true aspects, profoundly. By keeping the view of a Sustainable Development Goal 2 and 13, if you agree with me, to move ahead to catch the food security by keeping zero hunger and climate actions.
Right.
Will you put some glimpses? Already you have given detailed descriptions related how to we can achieve the food security, and this FDP play important role to make the policies henceforth. So we are very much in conjunctions with your talk that we have to work on the SDG 2 and SDG 13. Do you agree with my remark upon your presentation, sir?
Sure. Thank you very much.
Thank you.
So there's a question in the chat box. So the question is, sir, could you compare the Italian and Indian agro ecosystems, especially concerning the climatic change? So one of the participants is asking the question. Could you compare the Italian and Indian agro ecosystems, especially concerning the climatic change?
Well, very difficult to compare because the size of the dimension of the country is so different, and climate also. Although we have 1,000 kilometers from north to south, that is nothing in comparison to India. However, we are experiencing quite a lot of drought, quite a lot of floods. Climate change is bringing a change in the intensity, also in the intensity of precipitations, and with the losses for certain crops, important losses for certain crops, at least. As I not said, I have expanded on crops, not on animals or other things, because I want to concentrate in the first source of the commodities that supply. I mean, transformation by animals is already a transformation of what agriculture produces. The situation in Italy is that in the north you have the Alps and the Po River. In the south, we are starting cultivating cotton and banana. So that is the situation in Italy at present. I understand that is a normal thing in India because the country is so large, so long, difficult to make a comparison. However, the points I've mentioned are a reality in Italy as well. Some of them have been experienced. I refer, when I was talking about social economic aspects, I was referring mainly to what occurred in Italy after World War II, when the mechanization, new varieties and mechanization occurred, and the labor was available for industry. And the country has been industrialized by moving people from from villages to cities, and from agriculture to industry. That is, I don't know what is the situation, exactly what is the situation in India. The number of farmers in India are still very relevant. I feel that a number of them will move to some industry. An important point is to create industries that process agricultural commodities, both plants and animal commodities. A point I was also considering is that that will create difficulties. I was attending a meeting, a crop science conference, international crop science conference in New Delhi years ago, and one of the speakers, an Indian speaker, an economist, mentioned that we have too many farmers. 1/3 of them should disappear, should move to some other activity. And the Prime Minister, who was in attendance, just commented, "What shall I do with those?" That is what I expect to occur in India. In Italy, we are importing, and we need to import from North Africa, from India, from Bangladesh, from Pakistan, etc. People working on agriculture. And that is a problem, once a social program, but that is also an agricultural problem. I hope, my wish is that India will prepare policies to avoid what we have experienced, the negative aspects of our experience.
Okay, Professor, thank you so much.
So
Actually,
there's
how
one.
do you compare the minor cereals compared to major cereals? For example, you are also in Italy, durum wheat and other millets also. You are working and you are also interested in teff, Ethiopian crop also. So how do you compare the situation with these minor cereals, monocropping, how to overcome the monocropping to have the nutritional value? Can you please briefly describe it?
From nutritional point of view,
Yeah.
we have a pasta, still
Yeah.
a major importance on meals. But when I was a child, we had meat once, probably twice, some families twice a week. Once a week, say, during Mondays. All the other days were not, were just plant commodities. Now still pasta is an important food. But if the majority of families will serve meat at lunch and dinner. So the main component of proteins is no more plant products, but animal products. You had a terrible increase in one of the crops I've seen. Let me one of the slides. Eggs,
Yeah.
30 times what you had during 71, last century, 10 times meat, 12 times milk. That indicates clearly that India is moving on that direction. Crops will be important to provide feed for animals, not only for humans. And I think that with people moving to towns, working, husband and wife, time to cook is less and less, and they will move to prepared food. The point is to guide this change, not leave it to occur spontaneously or in disorder. That is a policy issue, but research should provide information on this. I don't know if I can see your point.
Thank you, Professor. Is there any questions from audience?
Yes, please. There are a number of questions. So I would read one or two, and rest I think I can we can send them to Enrico sir, and he can answer later due to the shortage of time. So one of the question is in recent year by Harikesh Singh. In recent years, crop residue burning has adverse consequences on environment. It elevates the soil temperature, thereby reducing the nitrogen and carbon. Can you suggest the remedies?
That is on tropical areas is an important aspect. One of the options is not the only one, but one of the options is the permanent crops. I've had experience in Central Africa, in Nigeria, and neighboring countries, Cameroon, etc., that a wind can able to move 20%, 20 centimeters of soil in a few days. So plowing the soil is a critical point on those. I don't know about the situation in India, but winds may be important. Plowing soil will move a certain part of it. That brings that soil is an important reserve of CO2. How to avoid plowing? Plowing is mobilizing soil, exposing to the atmosphere, and allowing CO2 to move to the atmosphere. So one point is, I feel, soil microbiology, crops which do not require plowing once a year, twice a year, on my assessment, are very important. I don't know if I answered to your point.
Thank you so much, sir. Thank you so much for the answering the questions as well. We still have some of the questions that have been asked by the participants, so we would mail the questions to you. So if you can answer them back, we can be the mediator and transfer the answers back. So due to shortage of time, we would like to move to the the second lecture. So I would
Thank
like to
you.
introduce.
Thank you
So
very
much.
much.
Yes, sir.
Thank you
Thank
so
you
much
very
for your
much.
time. Thank you so much. I would like to introduce our next speaker, Professor Birinchi Kumar Sarma, who is the professor, Department of Mycology and Plant Pathology in Banaras Hindu University. He is a distinguished plant pathologist known for his influential work in plant disease management, rhizosphere microbiome research, and host pathogen interaction in rice and pulse crops. He has completed his BSc from Assam Agriculture University and earned his MSc and PhD in mycology and plant pathology from Banaras Hindu University. He later pursued postdoctoral research at the University of California under the prestigious DST Boys Cast Fellowship in 2007-2008, a faculty member at BHU since 2002 and professor since 2017. Professor Sarma has made major contributions to understanding soil-plant microbe interactions, microbial ecology, and sustainable disease management. His work has strengthened eco-friendly crop protection strategies and advanced knowledge of the rice and pulse rhizosphere. He was elected a fellow of National Academy of Agricultural Sciences in 2024 and has received several honors, including the Best Teacher Award in BHU in 2013, NAS Associate in 2011, the Boy Scouts Fellowship in 2006, and the BHU Gold Medal in MS in 1997. He also received the Shastri Indo-Canadian Faculty Training Award for collaborative research at Dalhousie University 2018. Professor Sharma currently serves as editor of plant pathology for the British Society for Plant Pathology and is also a fellow of the Biotech Research Society of India. Through his extensive research, publications, and academic leadership, he continues to play a key role in advancing plant pathology and promoting sustainable agricultural practices in India. May I now kindly request Professor Sarma to please address the participants.
Very good morning to all. Thank you
morning.
for introducing me, and I am also thankful for the opportunity to share some of my thoughts with the audience. The topic that has been allotted to me is plant microbe synergy for climate resilient crop production. This is a topic that is very close to my heart, and my work is mostly centered around plant microbe interactions. And for last several years, I am also considering the climatic factors affecting this plant microbe synergy. So when we imagine a crop under field conditions, normally we talk about either a biotic stress or an abiotic stress, and we do a lot of work under that unidirection. But what is seen recently or in the past decade, that crops are being challenged by multiple stresses simultaneously. Say, for example, chickpea crop in Indian context, it is suffering from fusarium wilt, which is a very well-known fact. And this fusarium wilt can cause yield loss up to 100% depending on the severity of the plot where fusarium is prevailing. And at the same time, it has been seen that soil salinity is becoming a problem, and it is an increasing problem. And you can again see that extent of damage it can cause if the soil EC value goes up to 9 DSM, it can reduce yield up to 90%. So you imagine that a single factor which is quite capable of destroying the crop up to 100%, when these two factors combine, how these plants will survive. And more interestingly, under this particular complex scenario, what is the role of microbes or how they are actually contributing for the sustenance of these plants under these challenging conditions? That is what we tried to explore from our laboratory, and I will just present some of the facts, and we will try to elaborate that how these microbes are playing a significant role in this particular context. It is also shown that under soil salinity, or when the salinity level increases, the nitrogen use efficiency of the crops go down. That means a crop, whatever nitrogen fertilizer it is required, when the crop is under salinity-like stress, its utilization of this nitrogen is, again, it goes down much significantly. Here is an example of cotton. Here you can see that with the increase in soil salinity level, the utilization efficiency goes down from 40% to 25%. So this again shows that alarming situation that we are actually facing under field situations. And if you just see some of the current data in the sense that last year itself, Government of India has given subsidy to nitrogen fertilizers of around rupees 1.64 lakh crores. And in this financial year, it is estimated to be 1.84 lakh crores, but due to the problem with West Asia, this subsidy may go up to 3 lakh crores. So that means that already the government is under tremendous financial constraint because of this subsidy. And if the environmental factors like salinity and other things goes on increasing, this burden will also increase to even governments. So we wanted to explore a very potential microbe that is Trichoderma asperellum, strain T42, to see whether it has other role than protecting plants from biotic stresses like Fusarium wilt. So it is an established microbial strain that was helpful in protecting chickpea crops from Fusarium wilt. But we wanted to see what it is, how it is actually contributing to other factors of pulse crop, particularly with respect to nodulation, or if it is facing a simultaneous stress like salinity. So overall, a complex scenario, how this particular microbe behave. What we could see here is that this T42 strain Trichoderma, it did not increase nodule numbers in chickpea plant roots, but the nodule size were significantly enhanced, or the size become larger in presence of this particular Trichoderma strains in the rhizosphere, in addition to its normally established role, that is protection from Fusarium wilt and increase in biomass. So the data, when we collected, we could find that the biomass of the plants were increased when T42 was introduced. The nodule number did not increase, but nodule size were larger in comparison to the non-treated control plants. And the total nitrogen content is increased in chickpea plants, which actually attracted our attention towards that. Although the number of nodules were less in the roots, but the total nitrogen content were high in chickpea plants treated with T42. So we wanted to explore this particular phenomenon more under these challenging conditions, because as we have said that salinity reduces the nitrogen use efficiency, but introduction of a biological agent like Trichoderma, if it can enhance the nitrogen content under stress, even in salinity stress conditions. So this is something very interesting, and it may be a very, very useful technology that may be coming up, and we thought in that direction, and we tried to explore it further. So we just estimated the total nitrogen content, total protein content, and nitrogen use efficiency, and we have exposed the chickpea plants to single stress, combined stress, and then stress plus Trichoderma. So this was the experimental details given at the bottom. But here you can see in the right side in the histograms that most they were relatively higher in comparison to the left columns. And you can see that T starts from here. T is Trichoderma introduced. Here Trichoderma alone, Trichoderma plus pathogen, that is Fusarium, Trichoderma plus salinity, and Trichoderma plus pathogen plus salinity. So this you can see in the right columns. The columns height were relatively bigger or higher, in a sense that when we introduced Trichoderma, the nitrogen content, then protein content, as well as nitrogen use efficiency goes significantly higher in chickpea plants, even under stressful conditions. So that means one more thing that was established with this trichoderma strain, that it doesn't only protect plants, chickpea plants from Fusarium wilt, but it also contribute to the nodule health. Why we are saying nodule health? In a sense that because it does not increase the number of nodules, but the functioning of the nodule can be established through bigger size of the nodules and the nitrogen use efficiency data. So this is very interesting, and then we thought that we will explore it further in this particular direction. Then we wanted to also explore that once trichoderma was introduced into the soil of chickpea rhizosphere, what happens to the rhizospheric microbial community? So we adopted the metagenome approach, and then we found that there are some phyla that were abundance were more in comparison to high. But if we just consider the top 5 phyla of chickpea, what do you call that? Trichoderma introduced chickpea rhizosphere, there you can see that Proteobacteria, Acidobacteria, Bacteroides, Chloroflexi, and Cran archaeota. These were the top 5 phyla. Their abundance was higher in comparison to others. Then we wanted to dig more into the data, and we wanted to see what were the top 10, say, genus that was present in this chickpea rhizosphere. And we found that one particular interesting data, in a sense that you can see this green column portions here with red boundaries. These are increase in trichoderma plus pathogen and trichoderma plus salinity in comparison control, and these were nothing but the candidatus nitrosophora. And this is the one of the major dominant archaea species that was predominant in chickpea rhizosphere when trichoderma was introduced under stressful conditions. So we were not very familiar with this particular species, so we tried to find out what is the biological role of this particular nitrosophora. Then we found that nitrosophora were actually ammonia oxidizing archaea. They convert ammonia to nitrate, and then nitrate can be taken up by plants. So we just then, for taking up this available nitrate by plants, what is required is that plant uptake this available form of nitrogen through roots, through mediation or activation of certain genes, which we call it as NRT genes or nitrogen uptake genes. So it's in chickpea, they were not established how many of those were present. So what we did is that we took help of the Arabidopsis database, and then we tried to identify and find out how many chickpea nitrogen transporter genes were present, and then we took out everything from the database. And then when we went ahead and we just tried to see what was happening with these nitrogen uptake genes, we found that when trichoderma was introduced, many of the important nitrate transporter genes were upregulated by twice or even by 4 times in certain cases. And interestingly, the nodulation pathway genes or symbiotic signaling pathway genes, many of them which are mostly dominant in chickpea, were downregulated. That means when we have introduced trichoderma, it has helped the chickpea plants to activate its nitrogen transporter genes, which has probably been activated due to availability of available form of nitrogen in the form of nitrate. And this conversion of ammonia to nitrate probably done by the abundance of archaea species like nitrosophora. So this is the link that we could establish through the data that has come up from our experiments. And this is a very, very new way of looking into the trichoderma-mediated nitrogen economy in crop fields under stressful conditions. So we have published this particular part in the Plant and Soil Journal, and this is something to be looked upon very deeply in time to come. Then we also tried to find out that if there is any chickpea genes that is having some effect on nodulation activity or more nodule health related activities. We have taken the homology survey from Medicago species, and then we have found that one of the such nodulation related gene, that is Medicago BHLH1, this is a transcription factor gene, and chickpea has two homologs of this MTBHLH1, that is chickpea BHLS 114 and chickpea BHLS 115. So we wanted to see their role under these particular stressful conditions or different treatments. We have exploited three chickpea genotypes where having various degrees of resistance towards the Fusarium wilt. And then we tried to see how this Medicago homolog chickpea BHLS genes 114 and 115 performs under different treatments. So in the top panel, you can see that where there was no trichoderma, the expression were enhanced, but the expression were more of the same genes when trichoderma was introduced. That means this nodulation health related activities that we have predicted in our previous data that also helped probably nitrogen use efficiency. So nodule activity, enhancement and nodule activity could also be now relate with enhancement of activities of these two genes, that is BHLS 114 and 115, particularly 114, because its activity increased manyfold when trichoderma was introduced. And probably that is the reason we could see that the nodule size were larger in trichoderma introduced plants, even under stressful conditions. And probably this is one of those many genes that has contributed to the nodule health of chickpea and influenced by the trichoderma strain, that is T42. So this is the type of nodule status that we have already discussed earlier. Then we wanted to explore the entire family of chickpea BHLS genes, and we wanted to see how many of these genes are actually stress responsive genes. So we have found that there are 197 chickpea BHLS genes that are present in the chickpea genome, and 28 of them are stress responsive. So we wanted to explore this, and we have found that when trichoderma was not present, these 28 genes, they express differently under different treatments. But interestingly, when there was combined stress, that is PS means the pathogen and salinity, most of the genes were again downregulated under the combined stress. But to contrast, in the right panel, you can see that there are more darker spots that can relate to higher expression level of the same genes under trichoderma treated plants. So that means those 28 genes which we have identified as stress responsive genes, they are also expressed, their expression were also increased in trichoderma treated plants. Particularly, there are three genes which became our more interest to us because their expression was very high even in the combined stress of pathogen and salinity. So we wanted to explore further, and we just look into the sequences of the gene, and we have found that all these three genes has one commonality, that is they have an additional domain that is BHLS MIC N. So they probably this domain in these three genes might be having an important role to play for in giving tolerance to chickpea plants under combined stress of both Fusarium and salinity. Further, we wanted to explore that how out of these three genes, we wanted to see that how these genes actually interact with these proteins interact with other proteins. Then we have found that BHLS 119 actually interacts with a MIB transcription factor 114 and WD40 protein. So this made our case more interesting, and then we just tried to look into this particular BHLS gene more closely, and we docked this particular protein 119 with the database, and then we found that it goes and hit the MIP 114, and there's this BHLS 119 and MIP 114, then this dual complex then go and bind with WD40 genes. That means it forms a triplet protein complex, and this triplet protein complex, when it was docked, we could find that it comes and bind with the E promoter region E box of the defense related gene, that is PAL2. So that means activation of these genes now could be theoretically linked with defense, in a sense that because in the combined stress, this particular 119 was activated in presence of trichoderma. Now we can predict that it forms a triplet protein complex. Then it goes and bind with the E box of the promoter region of the defense gene, that is PAL. So here in the schematic representation of the phenomenon, here you can see that this is the PAL gene, and this is the promoter region E box, and this triplet complex come and bind with this promoter region, and then the PAL gene activation takes place. So later on, where we just wanted to see whether activation of this gene PAL gene has anything to do with lignifications or not, because this is the initial gene of the lignin biosynthetic pathway. And we could find that there was enhanced expression of lignifications that took place upon activation of the PAL gene. So this is gave now very well established that now how actually this trichoderma induced PAL2 gene mediated by activation of BHLS 19 could lead to defense in chickpea through higher lignifications under combined stress. We have also published this data in Plant Physiology and Biochemistry very recently. Then we also wanted to explore that how the chickpea epigenome works under stress. Normally, we all know that how a gene is expressed when the promoter region, when a transcription factor comes and bind, it leads to transcription and then subsequently translation or synthesis of a protein. But if in the promoter region, particularly in the CPG island, if any C or cytosine is methylated, then binding of the transcription factor becomes restricted and expression of the gene can be hampered. So if we wanted to explore that particular phenomenon, and for the students, I just wanted to describe this particular technique a little bit, in a sense that here in the left side, you can see that this is a normal cytosine, but when there is a methyl group is added, that it becomes methyl cytosine. Now, imagine this is a normal sequence of a gene. There may be two Cs, one C may be methylated and another cytosine that may be non-methylated. If you treat this sequence with bisulfite, then what happens? The normal non-methylated cytosine will convert to uracil, but the cytosine that is methylated, it will remain as cytosine even after bisulfite treatment. So after that bisulfite conversion, if you go for PCR, and then the PCR product, if you go for sequencing, then what you will find is that this uracil, then you will find it in the form of a T, that is thymine, and whereas the original C that is methylated, it will again appear as C. That means any original C that has converted to T, that means it was non-methylated after bisulfite sequencing treatment, but any C cytosine that were originally methylated, it will remain as C. So this is the technique that is used for looking into the methylation and non-methylation pattern of any gene sequence in a particular condition. So we went ahead and we wanted to see that entire chickpea genome-wide methylation pattern, and we could found that in control, there was 69% methylation that was originally present. But when you see that it gets changed with different treatments, but when trichoderma is challenged with the, chickpea is challenged with the Fusarium wilt pathogen along with trichoderma, then methylation percentage becomes very high. That is 72.6%. So this is just to give you an idea that when you expose the plants to different stressful conditions, there will be certain changes and the degree of changes in trichoderma plus fusarium wilt combination was maximum that we could found through our data. So we wanted to dig further with that particular phenomenon. We have already exploited the BHLS gene family. So we wanted to explore further. So out of the 28 stress responsive BHLS genes, we found that BHLS 92, it is having a CPG island in the promoter region. So then we have selected that particular gene promoter region, and then we have treated with bisulfite, and then sequenced after PCR amplification, we have sequenced, and then what we found is that there were 3 sites we identified where methylation and demethylation that took place. And all these 3 sites are actually MIB binding sites. In the BHLS 92, in the promoter region, we found 3 regions of CPG island where methylation and demethylation took place under different stressful conditions. But interestingly, all these 3 sites are MIB transcription factor binding site. So what we found in these particular sites 1 and 2 is that when we introduce trichoderma, here in the left, you can see that T and TP. Here you can see that these 2 became T. Originally, it was C. That means you can imagine that these were non-methylated C. And in trichoderma treatment alone and in combination with pathogen, this C got converted to T. The same thing happened here. Originally, it was C, that means non-methylated. And after bisulfite treatment, they become T, that means trichoderma has influenced them to become a T. That means they became demethylated. You can just imagine that they were C originally. That means they were methylated, and trichoderma caused demethylation, and that is why they got converted to T. This thing happened in these 2 sites. And in the site number 3, what happens? They were T mostly. That means they were originally they were C, non-methylated C. So after bisulfite sequencing, treatment and sequencing, they got converted to T. But in 2 particular cases, where S means salinity stressful conditions, they became C. That means they remained methylated. So under salt stress condition, they remain methylated, whereas in non-stress, non-methylated. So those, as I said, that BHLS 92 in salinity stress response, we wanted to see that how it is actually responding to salinity stress. So here you can see this BHLS 92, its expression is high, but in comparison to the same where we are indicating here is that it's a MIB binding site, and this MIB 78 is likely to bind in the promoter region of this particular BHLS 92. So expression of this MIB 78 becomes relatively less. That indicates that methylated status has inhibited this MIB binding activity to BHLS 92, and that is why expression of this gene were relatively less in comparison to BHLS 92. BHLS 92 is a salinity tolerance gene. It has expressed, and its promoter region CPG island 3 methylated status remain methylated, and that is why, because of that, it did not bind with MIB 78, and probably that is the reason that MIB 78 expression was lower in comparison to BHLS 92. So this is again another interplay between different stresses involving trichoderma and non-trichoderma and salinity stress. So this has given us again a very good insight that how epigenetics play an important role in stress tolerance, and many cases mediated by certain microbes. Here in our case, it is trichoderma. So here again, these are some related publications, not from our lab, but I just brought this particular publications into the discussion is that here people have also showed that under uh, stressful conditions, plant respond to certain stress like high heat or ROS generation is neutralized through root-mediated microbes through epigenetics. Similarly, the role of microbe is considered to be actually underused. That is the message from this particular communication, Nature publication last year. The authors here, they have said that how these microbes can actually help us to achieve one health that the whole world is trying to achieve. That is health of soil, health of crop, and human health. So all three can be actually accomplished through microbe interactions and plant microbe interactions. And this particular phenomenon is, according to them, is underutilized. And they are also advocating in the sense that if we want to actually achieve one health status, we have to actually go for utilization of microbes much more than what we are doing today. So overall, our work has also given some new insights, and the work from other laboratories across the world has advocated the use of microbe more and more, and it can actually lead us to sustainability and achieve the broader goal, that is One Health. So considering the time constraints, I will restrict my talk at this particular point, and I'll be happy to take any questions if any one of you have. Thank you so much once again for giving me the opportunity.
Thank you so much, sir, for an exceptionally informative and technically enriching presentation. Now I request the participants to kindly post their questions in the chat box.
You can also ask directly.
Dr. Sarma, hi.
Hello, Dr. Chaudhary.
Hi, how are you, sir?
Yeah, we're doing good.
Yeah. Nice presentation, sir, and provided glimpses on soil microbe synergy relate with the chickpea as well. As we know that trichoderma is highly used for biocontrol activity. You know that every child, every research scholars think in that way.
Right.
You provided some glimpses, very nice glimpses that it may helps not only in biocontrol, but as a, I can say the bioinoculant or the biofertilizer, whatever word you would agree. Very nice,
Yeah.
sir. And certainly you provided glimpses on not only the constitutive genes, but some other inducible genes also that are very interactive sessions. And in toto, how does the trichoderma, do you see some structural and functional divergence also? Do trichoderma affect other microbial populations, sir, in the rhizosphere on chickpea?
Yeah, that is actually that is another point that we wanted to discuss and bring out from our experiments. That so trichoderma is known for biocontrol. Trichoderma is known for nitrogen use efficiency, but it was not very known that how actually it is increasing nitrogen use efficiency.
Right.
So here we have established a role in nodule health. Its a role in nitrogen transporter genes activation. But the question arises why nitrogen transporter genes will be activated, because in nature nothing is activated for no reason. So here what we have established is that introduction of trichoderma actually engineered the rhizosphere soil under salinity stress condition particularly, and the halophilic archaea population becomes higher.
Okay.
And many of them, and you can see that one of the top archaea species genus is Nitrosophera.
Okay.
And its biological role is it is an ammonia oxidizing archaea. So it is converting ammonia to nitrate. So there will be available form of nitrogen around the chickpea plants, and
Right.
probably because of that, in presence of trichoderma, this NRT genes got activated much more because the nitrogen available form of nitrogen is present around. And it has uptaken this available nitrogen and then converted to protein
Nitrate.
and
Yeah, certainly. Right.
Professor Forchedu, please, you can ask your questions if you have. Professor Porchedu.
Unmute.
Unmute.
Thank you. Thank you, and congratulations for your presentation.
Thank you, sir.
I have a specific point. Any comment on soil microbia in during plowing, during soil disturbance promoted by agriculture?
That is
Soil as such and soil under cultivation.
Yes, yes. That is an area I think we have not actually worked on, but we are working on specifically identifying novel roles of one particular species, that is Trichoderma. As we can see that it is engineering the soil microbiome into some extent that can be linked with nitrogen economy of the soil in a stressful condition. But definitely when the soil is disturbed, the structure of microbiome get also disturbed. So I don't have any data to actually look into it and tell you something at this point. But I am sure that it's get disturbed, and introduction of that is why what we are doing also, that we are actually delivering this microbe through seed treatment. So that means whenever the chickpea seed is going to any part of soil, whether it is disturbed or undisturbed, our microbe is introduced into the rhizosphere through seed coating. So that is the approach that we have been adopting, and the results that we are finding under multiple stress conditions is being very, very encouraging to us, because chickpea otherwise nodule health goes down under salinity stress condition, its productivity goes down. But when we are introducing chickpea with microbes like Trichoderma, it is somehow reversing that particular phenomenon. So that is very, very interesting to us. And we thought that this is one likely technology that can go to farmers if it is established well.
Thanks.
Yes. Thank you, sir.
Thank you so much, sir. And there's one question from Mr. Sarvesh Singh. He's asking whether there is any role of trichoderma in increasing the rooting of the cuttings of the plant.
Definitely. Trichoderma, it is a very well-established role. Most of the Trichoderma strains, it encourages root biomass as well. So this is another mechanism through which actually plants can survive stressful conditions, including drought. So more roots means a robust root system will always lead to a healthy status of the plant, even under stressful conditions like high temperature or drought-like situations. So there are reports, and Trichoderma in our strain also, where this is very well evident that it enhances the root biomass. Yeah.
Thank you so much, sir. I hope the sir has answered the questions now. So thank you once again, sir, for a wonderful presentation. It
Thank you
was
so
very
much.
informative, very informative. Thank you so much.
Thank you so much.
Thank you so much. Our next speaker, Dr. BP Mallikarjuna Swamy, is a senior scientist in rice breeding and biofortification in International Rice Research Institute. He is an internationally recognized rice geneticist and breeder known for his outstanding contributions to rice breeding, biofortification, drought tolerance, and climate resilient agriculture. Born into a farming family in India, he developed a deep interest in agriculture and farmer welfare from an early age. His passion for improving the livelihoods of farming communities inspired him to pursue a career in agricultural sciences and plant breeding research. Dr. Swamy completed his doctoral degree in genetics in India in 2009. His PhD research led to the identification and mapping of major quantitative trait loci associated with grain yield and related agronomic traits in rice. His work also resulted in the registration of novel germplasm, and the release of high-yielding rice variety, reflecting his early scientific excellence and practical contributions to crop development. Driven by challenges posed by climatic change, food insecurity, and global malnutrition, Dr. Swami joined the International Rice Research Institute in 2009 as postdoctoral fellow in drought molecular breeding. During this period, he successfully mapped major QTLs for grain yield under drought stress and pyramided these QTLs into popular rice cultivars together with other important loci for biotic and abiotic stress resistance. Recognizing his scientific achievements and leadership potential, IRRI appointed Dr. Swami as lead breeder for healthier rice development in 2013. Since then, he has played a pivotal role in the development of nutritionally enriched rice varieties, including contributions towards the advancements of golden rice and micronutrient-rich rice cultivars aimed at combating vitamin and mineral deficiencies in vulnerable populations. His research integrates advanced molecular breeding, genomics, and biofortification strategies to enhance both nutritional quality and climate resilience in rice. Dr. Swami is also a prolific scientific author, and he has numerous publications in high impact international journals. He actively contributes to scientific capacity building through training, mentoring, dissemination of knowledge related to plant breeding, nutrition, and sustainable agriculture. In addition, he serves as an active member of several plant science societies and editorial boards of scientific journals. With his strong commitment to science-driven agriculture innovation and global food security, Dr. B.P. Mallikarjuna Swamy continues to make scientific contribution towards the development of healthier, resilient, and farmer-friendly rice varieties for the future. Now, I invite Dr. Swamy for a much-awaited lecture. Dr. Karthikeyan, can you please upload the presentation from your end? There might be an option of share.
Deepshika, Dr. Karthik has uploaded. Please move.
Just go back one slide.
Yeah. Yes.
Okay, so can I start?
Yes, why not?
Sir, good morning,
Yes,
please.
sir, sure.
Yeah. Thank you very much for this invite, and also I would like to congratulate Amity University of Agriculture for organizing this 5 days program on net zero emission agriculture, and also thanks for this opportunity. So myself, I'm Dr. Swami. I am with IRI for almost 17 years. So, and also thanks for your introduction. So today I will be giving you some glimpses, some highlights of our work on development of healthier rice to achieve global nutrition security, and that's the title of my today's presentation. So next, yeah. So
Here it is mute, no? Audio.
Yes.
Sure.
can you go to full window mode? Hello. Yeah, next slide. Yeah. Yeah. So, as you all know, that rice is an important staple food crop for almost 50% of the world's population, and we eat rice on everyday basis for our energy needs as well as our nutritional needs. So, rice plays an important role in food and nutritional security, especially in Asia, where more than 90% of the rice is produced and consumed. And as we all know that population is growing and burgeoning population that is going to reach almost 8.5 billion by 2030. And also this most of the increase happens in Asia and Africa. And we will be flooded with the challenges of producing more with less land, water, and less resources in order to secure food for our people. Not only food as well as nutrition. Rice, as of now, it's grown on almost 150 million hectares with an annual production of 6 million tons. And even the rice production is taking up, is going in a bigger scale in African and Latin American countries. So with the economies increase, their economies are doing well and people want to eat rice. So even in African countries and Latin American countries, there's a huge demand for rice. So as I said, so we need to produce more and we have to make it more profitable because people say that rice cultivation is costlier. So we need to invest more. Profit margins are less. On the other side, since people eat almost 3 to 5 times a day rice, in some of the Asian countries they eat even 5 times a day. So and the rice that we eat nowadays is polished. So most of the nutrients are removed and also even the high yielding varieties that are produced during Green Revolution are less nutritious. So the thing is that we are eating just food, rice only for energy. So it doesn't provide much the nutrition that we need on everyday basis. Just to give you an analogy, suppose you have a vehicle, just you put only petrol and diesel, you don't do any service or put grease and other stuff to maintain the different parts, it will not be long lasting. Similarly, if there are no nutrients, only energy, people face a lot of health issues. So nutrition is very important. Earlier, people used to get most of the nutrition through the food they were eating. But all of the practices and all the new varieties that we have bred and the post-harvest practices, they have a impact on the nutritional status. So the challenge is that we need to produce high yielding and nutritious varieties and that are affordable and accessible at all the times. And also that's an important sustainable development goal to provide food and nutritionally important or high yielding crop varieties of major staple food crops. Next. So if you look at globally, some of the major top 10 health and nutritional issues, blindness, stunting, anemia, these are some of the major problems. And health issues like diabetes, cardiovascular diseases, stress-related issues, and heavy metal toxicity. So these are some of the major top 10 nutritional health issues that are plaguing the world. And especially children, pregnant and lactating women are highly prone to micronutrient deficiencies because they are in much need of these micronutrients for their special needs, growth and development. So they are, and even elderly people, they are highly prone to these micronutrient deficiencies. So there are many approaches to provide nutrition. Among them, biofortification is also one of the approaches. So that people are considering and a lot of impact has been already made. So next slide. So on it, you can, as I mentioned, there are different approaches to get derive your nutrition on a daily basis. You can change your diet, or you can get supplementation tablets and pills, and through biofortification. Biofortification means the breeding process by which you will increase the nutritional content of the edible portions, and that should be bioavailable. That means it's not just enriching, that should be bioavailable when we eat it and should make available, and that should help in reducing your nutritional deficiencies. And it's one of the most cheapest approach so to provide nutrition, and almost 600 microgram of vitamin A, almost 15 milligram of iron, zinc, needed on a daily basis. But most of the modern rice varieties, especially in their polished form, they are less nutritious, almost 2 to 3 ppm of iron, 12 to 16 ppm of zinc, and there is no variability for vitamin A in rice. So these amounts of nutrients are not enough to meet the daily requirement of these vitamins and minerals. So next. So biofortification of rice varieties is the most go back, please. Biofortification of rice varieties is the most sustainable, targeted, and a cost-effective approach to alleviate the problems of micronutrient deficiencies. Because if some people have to take supplements or tablets every day, nutrient supplements, they have to invest every day. And even there is approach called food fortification. So even for food fortification, there's a lot of investment on a regular basis. They are not sustainable. But if you develop nutritious varieties, that's more sustainable. Once you develop and release it, people can cultivate it and they can consume it and they can derive nutritional benefits. So another important thing is that the food matrix plays an important role. So if the nutrient is available in the food matrix, means the rice that we eat and we derive most of the nutrition. If you provide nutrition in the staple food, that's readily available for the body. So that will be more impactful to reduce health impacts. Yeah, next one. So at TRA, we're working on developing high zinc rice by breeding, high development of iron zinc rice by transgenics, and golden rice, and also there's work going on diabetic rice, or even there's a lot of work nowadays on traditional land races to assess their health and nutritional benefits, and lot of value addition is happening. And also dietary diversification, we are always advocating wherever we are going and meeting people. We are also advocating dietary diversification as well. Next one, please. So next, if you look at a little bit in depth on importance of zinc for human health and nutritional aspects. So zinc is very, very important because for more than 300 enzymes and more than 3,000 proteins, zinc is must. So even all our transcription, translation, most of the activities, it's an important cofactor. So that's why even for the absorption of iron, zinc is must. If there is not much zinc, how much amount of iron you take, it will not be available for the body because it's a major cofactor for activities of enzymes and also for the functioning of the more than 3,000 proteins. That's why whenever people go for anemia, doctor will give you iron and zinc supplements together. They will have a syrup that has both iron and zinc. So they have to go, it has to be given together. So I'm looking at the amount of rice eaten on a daily basis and amount of zinc that is retained after washing, cooking, and how much is retained. So they have set a target. Like the aim is to provide 36% of the estimated average requirement. And as I mentioned, the current varieties have 12 to 16 ppm. The target is to increase it by additional 6 to, go back, please. Additional 6 to 12 ppm. So on an average, 5 to 13 ppm of zinc is required depending on the age, gender, and growth stages. So the target is to increase the zinc content by 6 to 8 ppm in the first wave of varieties and 8 to 12 in the second wave of varieties and almost more than 12 ppm over the basal 12 to 16 ppm in the third wave of varieties. We are already there in the third wave of varietal release and almost 18 varieties have been released in different parts of the world. Yeah, next one. And if you look at the distribution of iron and zinc in the rice grain, so almost 60% of the zinc is retained in the endosperm. That means even after polishing, there is enough variability for zinc in the rice grain. But on the other hand, most of the iron is in the outer layer that gets removed, or we will lose that portion because most of the iron is in the outer layer. So there is not much variability for iron, but there is variability for zinc in the endosperm. So breeding is the best possible approach for zinc, improving the zinc content. For iron, genetic engineering or the genome editing approach is being followed to increase the iron content in rice. Next one, please. And we follow molecular approaches and conventional breeding approaches to increase the grain zinc content in rice. So we identify high zinc donor lines with acceptable yield potential. We do all the breeding works like any other standard breeding programs. We make crosses, we take through the different trials and get nominated. At the same time, we also do a lot of QTLs and genes identification, integration, and even transgenic approaches to come up with conventionally bred as well as molecularly bred or even transgenic high zinc rice varieties. Next one, please. And as I was mentioning, the land races, they are rich in micronutrients, but they are low in yield. But ultimately, when we have to have a variety, for a farmer, yield is the most prominent one thing. So if the variety is not yielding, how much amount of nutrients are there, no farmer is going to grow. So the thing is that we need to have these nutritional rich varieties in the yielding backgrounds. They should be comparable or even better than the existing varieties for their adoption. So in rice germplasm collection, we have different subgroups called indica, japonica, house and aromatic. Indica are the ones that we eat on a daily basis. So they are the lowest in terms of nutrition compared to japonica, compared to house and aromatic. But these house and aromatic are genetically closer to indica. So in ERI gene bank, there were 3,000 accessions. So I tried to identify some of the accessions with high zinc content as well as a yield. So we identified some of the very interesting donor lines for improving the zinc contents, which are acceptable in terms of yield potential as well as high in zinc content. And we use these lines for all molecular work as well as for developing high zinc rice varieties. Next one, please. Next slide, please. Yeah, so go back. Yeah. Zinc is highly complex, genetically highly complex. Yield is also highly complex. So we are handling too many complex things. So and also there is a dilution and concentration effects. Suppose if the yield is high, the zinc level goes down, and if it is yield is low, the concentration of the zinc goes up. So we need to handle this. As I said, we need to increase the concentration of zinc in a yielding variety background. That means yield should be high and zinc should be high. And sometimes people say, why don't you apply zinc to the soil so it will increase the zinc content? That's not true. For example, zinc deficiency in the soil, it may be artificial or it may be natural. So artificial means there is a lot of zinc available in the soil, but plants cannot take it up due to its complex nature, its unavailable form. On the other hand, natural zinc deficiency, there is no zinc in the soil. So under artificial zinc deficiency conditions, only the genotypes that have a capacity, they can produce a lot of root exudate, they take it up, load it, and they can put it into the grain. But under natural zinc deficiency, we need to apply zinc to the soil. And soil application versus foliar spray. As I mentioned, any amount of zinc application under artificial zinc conditions doesn't make any difference in the grain zinc content. But on the other hand, foliar spray will increase our 2 to 3 ppm. And remobilization versus yield penalty, even there is almost more than 300% more zinc in the vegetative tissue, it won't translocate everything into the grain because if there is a lot of remobilization that turns leaf into yellow, that leads to yield penalty. So these are some physiological bases. So we are working on that to how to balance that. And even irrigation, like irrigated, rain-fed, and alternative wetting, drying, they will have an impact. Like a lot of environmental factors also will have an impact. So zinc deficiency operates at the system level, means if there is a zinc deficiency in the soil, the plants and the food that we produce are also zinc deficit. And people who eat such foods, they are also zinc deficit. That means it goes from soil to the plant to the food that we eat and to the human beings. So that's how it flows in the system. Next one, please. And we did some experiments in the Philippines. We collected a lot of data from the Philippine Soil Science Bureau and human nutrition data and from the rice databases. Then we overlapped all this data using GIS tools. It was very clear that wherever there is a soil zinc deficiency, the rice produced in those areas had a zinc deficit grains. And the people, those who are eating foods from such areas, also zinc deficit. That means, as I mentioned earlier, it operates at the system level. Yeah. Next one, please. And as I mentioned, biofortification is not just increasing the bioavailable grain zinc content, and it also includes increasing the promoter compounds, reducing the concentration of anti-nutrients, and even improving the nutrient content through agronomic approaches. These are all comes under the definition of biofortification. And if you have to have a variety successfully bred for nutrition, it should be high yielding, stress tolerant to multiple biotic abiotic stresses. Along with that, we need to provide nutrition. Then only it will be successful in the farmer's field. And these are some of the high zinc donor lines that we identified from our subgroup. Their nutrient levels are also high and they have acceptable yield potential. Next one, please. And we also did a lot of molecular work, like we have developed multiple populations, GWAS and biparental populations, a lot of in silico analysis to identify genes and QTLs, and developed SNPs for using in LDR rice breeding programs. Next one, please. After quite a lot of studies using multiple populations, multiple locations, multiple years, we were able to identify two very, very consistent QTLs. Zinc 7.1, on chromosome number 7, and 5.1 on chromosome number 5. Very interestingly, these were co-located with very important genes which are involved in zinc homeostasis. For example, zinc 7.1, it was co-located with the nicotinamide synthase gene. That's a well-known chelator transporter and also helps in accumulation in the grain. And zinc 5.1 is a transcription factor that helps the activation of WOSNAS3, all the WOSNAS genes. Yeah, nicotinamide synthase genes. Next one, please. Interestingly, yeah, next one. So it's not just two studies. You can see multiple populations, biparental, multiparental, magic populations. The QTLs are very, very consistent. These 7.1 and 5.1. Only such major effect and consistent QTLs are useful for breeding. Next one, please. Yeah. And interestingly, we wanted to look at the relationship between these 7.1 and 5.1 QTL. And as I mentioned earlier, 5.1 is co-located with the transcription factor GIP5 and GIP9. And the 7.1 was co-located with the nicotinamide synthase gene. And when we looked at the transcription factors, what are the immediate genes that are being influenced by this transcription factor? Interestingly, nicotinamide synthase 2, 3, and 1, they are the genes which were influenced by this transcription factor GIP5 and GIP9. That means during the breeding for elite varieties during the Green Revolution, most of these important alleles of these interesting genes have been lost. So they always selected weak genes because there was no much focus on nutritional improvement, only yield. So we lost some of the important genes for nutritional traits. Yeah, next one, please. So we also did some QTL effect, pyramiding effects, and you can see yourself as the number of zinc QTL increases, the additive effect of the QTLs also increases. You can see the lines with the 4 QTLs had highest zinc content, almost 6 ppm. Even we looked at some of the haplotype effects. Some of the haplotypes are more prominent and useful for zinc biofortification. So now we have almost 3,000 genome sequenced. It's not only QTLs and genes. We can also look at some of the most efficient alleles or the haplotypes for those genes. So we did that for this QTL 6.217 and some of these, for example, the haplotype 80 is much better than CC. Similarly, CAG TTT haplotype is much better than the other one for the QTL zinc 7.1, the gene post nicotinamide synthase gene. Yeah, next one, please. And we did meta-analysis from all the available literature. So, and we identified 52 meta QTLs for zinc. From there, we shortlisted 18 meta homeostasis genes. Next one, please. Next slide, please. Very interestingly, you look at if you have a look at these, so we identified different haplotypes for these genes and their efficient haplotype forms. Not only that, after identifying these haplotypes, we validated their phenotypic performance in the field. So we got very interesting results. So these haplotypes and the 3000 genome information is highly valuable in terms of identifying the genes and their haplotypes, what kind of haplotypes we should focus, and we should use it for SNP development and mobilize them into the rice varieties. And also we looked at where these genes are getting expressed. They are expressed in roots, in flag leaf, and in the grain, in the panicles. So it matches with their role in zinc homeostasis in the plant. Next one, please. So we nowadays there's a concept called zinc mainstreaming. That means earlier people a lot of focus on only yield. So now people wants to put a threshold on the nutrition. That means if any variety has to go through the varietal release process in any country, they should have at least some minimum threshold for the nutritional content. So these things are coming up and they are becoming policy issues in most of the countries. And in the coming years, you will be seeing such kind of conditions put in place to get the varieties released, because nutrition is very, very important. Yeah, next one, please. And we are running this program quite a lot of number of countries in Asia, Africa, Southeast Asia, South Asia. So these are some of the lines and the countries that where we are focusing our work. Next one, please. And for varietal release, there's a lot of criteria. So there's some different countries, they have their own criteria. So this is the criteria we came up. I suggested it to the authorities in different countries that they release varieties. So as I mentioned earlier, there is a dilution effect and concentration effect. Earlier, they were using only zinc differential. That means the current variety and the new variety that we are going to release, there should be a minimum of 6 ppm difference. But that's not enough. We need to look at their mean performance, zinc differential, as well as absolute zinc content if you have to recommend a variety for release. So this is the one which has been well taken by all the policymakers in different countries, and they are positively implementing it. Yeah, next one, please. And vitamin A deficiency, as you all know, vitamin A is very important for vision because it's a component of retinol. So almost 190 million children, 19 million pregnant women are affected by vitamin A deficiency, and also is related to the immunity related stuff. So, but there is no variability in the rice. So they incorporated a gene from maize and soil bacteria, and they developed a variety, golden rice variety in the background of Cabonet, which is a US based variety. So that was imported to Erie way back in 2007. So we introduced that gene into popular rice varieties, and next one, please. And we also successfully got food and feed safety approval, and it was successfully commercialized. But as you all know the story, again, this Greenpeace and Masipak, they went to the court. There is some temporary restriction for its deployment. So we incorporate this gene into multiple varieties from Asia, and the product was really good and it was going very well until the Greenpeace came in and we got some temporary restrictions on deployment. There was a lot of interest and support from the lot of stakeholders, but ultimately, for time being, now it's temporarily stopped from deployment. But very soon we'll be hearing some good news on that. Next one, please. And people always ask these lot of questions like how it tastes, how it smells. So there are a lot of studies we did, composition analysis, they taste, smell, and everything is same, but only the difference is that improvement in beta carotene content. Next. Yeah, this is one of the secretaries or the minister in the Philippine government. You can see, and also ambassador from US, they are tasting golden rice. So it tastes, smell, just like any other ordinary rice. Even they say even it tastes much better. Yeah. Next. So golden rice is safe, stable, and it can be cultivable, and it provides you enough nutrition. And it has been successfully deregulated and it was released, but now there is a temporary ban. Next one, please. So similarly for iron rice as well, so using different strategies or genes for different strategies like increase in iron storage capacity and translocation, and those genes involved in either storage or translocation or combination of those have been identified and transgenes have been developed and tested. And you might have seen the news last week that there is a commercial propagation approval from the Philippines to cultivate. People now can cultivate the high zinc rice varieties as well. That's transgenic. Yeah. High iron and high zinc transgenics. Next one. So these are some of the field trials. You can see the nicotiniamine synthase and ferritin genes. The trials conducted at Erie and CRT in Colombia. So you can see yourself, they have much more higher levels of iron and zinc, almost 50 ppm of zinc and almost 15 ppm of iron. So it shows the very high increase in the nutritional content. That's not possible through the conventional or molecular breeding. So transgenic has some advantage where there are no alleles or the genes available in the native gene pool. So this is an evidence for that. Next one, please. So these are now we wanted to stack iron, vitamin A and zinc. So that work is in progress and we have already lines have been developed and tested. So we need to provide multi-nutrient varieties. So with high iron and zinc and vitamin A. That's the project that's under progress. Next one, please. So during Green Revolution in 1960s, you might have heard this slogan by Norman Borlaug, food is the moral right of all who are born into this world. But to a major extent, food problems have been solved. Now we can say that nutritious food is the moral right of all who are born into this world. I think the efforts are globally going on in that direction. It's not just food, it's the nutritious food. So there's a lot of awareness and the demand for nutritious food worldwide. Next one, please. I would like to thank all the donors and the participants and the team members, and also I would like to once again thank Committee University of Organic Agriculture for this opportunity. Thank you so much once again, and wish you all the best with the rest of your program.
Thank you so much, sir. Thank you so much for an excellent, engaging, and technically stimulating presentation. Now, I would request the participants to type their any queries or questions in the chat box, please. And the faculty members can ask as well.
Dr. Karthik.
Sir, how you can overcome the yield penalty? For example, mostly zinc and yield can be correlated negatively, as you also explained. Did you observe any specific variety apart from the general case? Do you have any specific variety to compensate this problem?
Yeah, so actually that's what exactly I was talking. Like most of the times, the biofortification breeding program, just they randomly choose the donor lines. So as I was mentioning to you, always there are exceptions in the nature. So that's why we screened more than 3,000 house accessions in the gene bank. From there, we selected 600. Again, we selected 40. We tested across the locations, and we were able to identify almost 10 to 15 donor lines which are having acceptable yield potential and high grain zinc content. Now you see most of the varieties that we have released, they have higher yield potential and they have grain zinc content because they have to go through the stringent selection criteria and release criteria. For example, in India, we have released two varieties, Spurti, and there is another one recommended just last month. They say that the yield is very good yield in the Spurti and as well as the second one. So if they don't cross the thresholds, it won't go. But what you said is right. Most of the times, that's how there is a negative relationship. But always nature has exception. That's what we breeders have to identify and use it in the breeding program. So selection of donors are very important, and we selected high zinc donor lines with acceptable yield potential and which are genetically closer to indica type, and we could able to develop varieties with high yield potential as well as high zinc content. Yeah.
Okay. Audience, do you have any questions?
There's a question from Medha Panthali. He is asking how much effective is zinc seed priming?
Yeah, so yeah, as I mentioned, the zinc seed priming definitely it helps, especially when there is these artificial zinc deficiency conditions. As I mentioned, that helps. That will help you to trigger some of the genes, so root growth and development. But that helps only in the vegetative establishment of the crop, your crop. But it won't help you in translocating or accumulating zinc in the grain. So that helps in your crop establishment and growth and giving you more yield. But that has nothing to do with the zinc accumulation in the edible parts.
So when you compare to wheat, you can see generally in wheat, it can accumulate even 35 ppm, 30, even 40, normally, in normal case. While in rice, it is generally very less compared to wheat. Maybe some
Yeah,
different gene expressions are there. Do you compare that in the future with your study?
yeah, that's very interesting. But if you look at the method of cultivation, so rice, you always flood the water, under flooded conditions or anaerobic conditions, zinc goes into complex form. So it will not be that readily available like in a wheat where you just grow in normal conditions. Now you don't flood the wheat field.
Maybe
That's
direct seeded rice can be useful.
in the direct seeded rice, yeah, it can reach up to the level that you are talking, but at the same time, your direct seeded rice won't give you the yield like under irrigated conditions. So there will be some amount of concentration effect there. So
Okay.
yeah, crops are different, and their morphology, roots, and all those things are different, and their cultivation methods are different. And since you flood a lot of irrigation and there is standing water, sometimes even the zinc that you apply, once it goes to the soil in the flooded conditions, immediately it converts into unavailable form. Plant has to produce a lot of fruit exudates and energy there to release it and take it up. So that's where the breeding comes in. You need to have genotypes having that much capacity, yeah, to take it up and put it. Yes.
Thank you for the answer, sir. Dr. Jimmy
Mr.
John
Gupta.
has asked.
Yeah,
Do
please.
you want to ask? Okay. So Dr. Jimmy John has asked, is the gene accession IDs based on Japonica rice or Indica rice? The accession IDs are based on Japonica rice.
These are all based on nowadays we have multiple genomes available. You can compare whatever you want. So you don't see much completely synteny and things are changed. So what we used is using the
Yeah.
indica one. Yeah, house and indica they're genetically closer. And for rice, as I mentioned, you have a 3,000 genome sequenced. Now your choice, whatever you want, which genome you would like to compare, which prototypes you want to use. So mostly because most of our countries we eat indica type of rice, and mostly we use always indica based genomes for comparison and the gene IDs.
Okay.
And there is also another question related to golden rice polishing. There is not much effect because as I mentioned, our focus of improving nutrition is always in the endosperm. So since it's in the endosperm, polishing will not have much effect in that.
Sir, DK, sir, you wanted to ask something?
Yeah, just Dr. Swami, thank you for
Yes.
such a very vast knowledge that you shared with us. Sir, I was wondering to find out zinc finger proteins already locate in the chromosomes or the constitutive genes in the rice, if I'm correct, right? So do outside zinc will mask the constitutive genes or it may try to help in induction of that gene? That is my just lay questions.
Yeah, as I mentioned, so it's not just one gene or a couple of genes because as I mentioned, it's the whole metal homeostasis. We call it a whole metal homeostasis from releasing zinc to the soil, taking it up, root take up, loading into the phloem, taking into the tissues, putting in the vegetative tissue, and it's a whole complex form. So hundreds of genes are involved. So there are some constitutively expressed, there are some which are induced. So if you look at a lot of studies available on expression basis, so there are some, there are reports, they are induced. When you apply gene, they get induced, and also under different stress conditions and combinations. So there are such like that. But there are also set of genes which have constitutive expression. That has to happen on a regular basis because it's a life process on a routine basis. So since zinc is important not only for us, even for the plants. So there are certain things happens on a daily basis. So for us, we want something that plant has to do something for us to put more nutrition into the grain. For plant, it may not be that much enough. It's a little bit is enough just to survive and grow, but we want more. So there we want to bring genes and express and put it into the grain. So it's a combination of both. Yeah.
Okay.
Yeah.
Thank you.
Thank you, sir.
Thank you very much. Thanks as well.
Thank you.
One last question, sir, from Dr. Devi. She's asking, given that black rice, which is known as chakao in Manipur state, has received significant attention for its antioxidant properties, are there any ongoing efforts to transfer these beneficial traits into high-yielding rice cultivars through breeding programs? Kindly share your insights.
Yeah. People are always trying these days, as I mentioned, a lot of attention on heirloom rice and all these traditional land races. But breeding, they have to have a separate interest and secure funding and a lot of interest there going on. But not sure the scale and the efforts. But I'm also working with Assam Agriculture University. They are my collaborator. So they are working on that aspects as well. But these are special products and derived products. They have to have breed in a different way and they have to come up with products and packaging and selling. So, but to make it mainstream, still quite a lot of time. But on the other way, you look at some of the intelligent people, some countries, they are much advanced. For example, European countries, they go to Thailand and they buy lot of colored rice with lot of they put lot of rules and regulations and procedures, and they earn multi-billion dollars. They produce food colorant, all medicinal compounds. It's a big business, multi-billion dollar business. So there is an opportunity. There's a lot of interest. So it all depends on the policies, procedures, and government priorities. But there is a plenty of market for land races and traditional varieties. Yes, please.
Thank you so much, sir. Thank you for your answers. With this, thank you so much for your lecture. So with this, we come to an end of day one, morning technical sessions of FDP. And on behalf of the organizing committee, I sincerely thank all our esteemed speakers and participants for their active engagement and valuable contributions. We shall now break for lunch. The next session will resume at 2:10 p.m. I request all the participants to kindly rejoin the platform.
To have my FDP is going on. This is my lunch break, and my FRC. Sorry,
I think ma'am is on.
SRC is also
Ma'am, please
going
mute
on.
yourself.
Tomorrow is DRC. Can we talk over phone?
Dhoni ma'am.
Once again, I would like to thank all the organizers. So I need to leave now. Thank you so much. All the best.
Okay, sir. Thank you so much.
Thank you so much, sir.
Thank you so much.
So we shall break for lunch now, and I request all the participants to please join the session few minutes prior to 2:10 so that we can start the next session on time as well. Thank you so much for joining, everyone. Thank you.
Okay, yeah, awesome. Video. Good afternoon, Dr. Patricia Galefi. Hello. Hello, ma'am.
Hello, can you hear me?
Hello, ma'am. Good afternoon. Are you hearing me?
Yes, I am
Yeah,
hearing.
I think you are in mute. Could you please unmute yourself? In your computer, you have muted. Could you please unmute yourself?
Oh, yes, okay.
We are not hearing your voice, ma'am. We are not able to hear your voice.
Dr. Karthikeyan, she is
I think
audible. We are
unmute.
able to hear
Could you please
her.
unmute.
Dr. Karthikeyan, we are able to hear her.
You have background?
I don't. Sir. It's okay, sir. It's not a thing.
Not a big issue.
Yeah.
Let me
I
speak. Let
think-
me speak with the practice. Yeah. No.
So you need to unmute. So you- Yes, we are not able to hear you.
Karthik, I don't understand. I don't hear you.
Patricia. Dr.
Okay, now.
Hi, this is Dr. D.K. Chaudhary. We
Okay.
welcome you over here through Dr. Karthik, and we requesting you please unmute yourself through Zoom or see if computer speaker is off. Please.
We are not
Okay.
able to hear your voice.
Yeah.
Sir, ma'am is audible.
DK, ma'am is audible. We are able to hear you. Probably you have issues in your connection. हाँ?
Separate हो जाते हैं।
Separate की बात नहीं है। अगर ये नहीं कर पा रहे तो give it to Dipshi. Don't waste time here. You don't have a LAN connection. आपको भी सुनाई नहीं दे रहा?
Ma'am, can you, I think from your side, we are not able to hear your voice.
May I start?
Can you talk, please? We can see.
May I start now?
Uh,
It's my time.
yeah, you can. Yes.
Good afternoon.
Yes.
Just a minute, please. Good afternoon, Dr. Patricia.
Yeah.
So to introduce our next speaker, that is speaker number 4, Dr. Patricia is a distinguished plant geneticist. Am I audible, please?
Yes, ma'am.
Okay. So Dr. Patricia is a distinguished plant geneticist and biotechnology researcher at ENEA Casatia Research Center, where she serves as a group leader in the Laboratory of Agri-Food Sustainability, Quality and Safety, renowned for her interdisciplinary contributions to plant molecular biology, crop genetics, and sustainable agriculture. Dr. Patricia has played a pivotal role in advancing research on drought tolerance, genetic diversity, and climate resilient crop improvement. Her scientific work integrates molecular genetics, biotechnology, and microbial resource management to support sustainable agri-food systems and bioeconomy initiatives. Dr. Jellife's research focuses extensively on understanding abiotic stress tolerance mechanisms in crop plants, particularly durum wheat and triticale. Her studies on stress-responsive genes, including the characterization of genes associated with drought resilience, have contributed significantly to the development of climate-smart agricultural strategies. She has also been actively involved in innovative research exploring bioethanol production from triticale lines, highlighting her commitment to renewable bioresources and sustainable agricultural technologies. As principal investigator and research leader, Dr. Jalifi has contributed to several major European and international projects, including PE10 on foods and dairy soy, Simba, Eco-Ready, etc. In addition, she serves as a scientific coordinator and curator for the ENEA Microbial Culture Connection, supporting microbial biodiversity conservation and its application in agriculture and biotechnology. With more than 65 peer-reviewed scientific publications, Dr. Jalafi is widely recognized for her impactful contributions to plant science and sustainable agriculture. She is also a respected member of Academia de Geographie, one of the world's oldest scientific agricultural academies dedicated to the advancements of agricultural sciences and environmental sustainability. Dr. Patricia, please welcome. We welcome you on behalf of Amity Institute of Organic Agriculture, and I would request you to please start with the lecture so that it is a much awaited lecture among the participants. So I think we can continue now if you're comfortable.
Okay, thank you very much.
Thank you.
Thank you. I started to share the presentation. Where is now? Okay. Just a moment, please. Okay. So you can hear me?
Yes, we are
Yes,
hearing.
thank you. So, first of all, I want to thank you very much, Kartikeyan, for this very, very important invitation. And I want to thank all your colleagues and your director, professor, because I think this workshop is very important very full of very interesting lectures and talks. So I am very honored to participate. I also want to say some words about that. I am just retired, so I want to talk also in general words. I mean, not the last research results, okay? Just something that is coming also from literature and other information around. Because the problem of a climate smart agriculture and the future of agriculture in the world is a very big problem. I mean, we know that in agriculture, many factors are contributing to the final harvest, to the final yields of every crop. And we know how much is important to have a potential yield in any crop and also the stability of the yield. And we know in several years of studies that the yields are changing every year. And many times the yields are affected from different factors that are much more linked to environment. I mean, the climate is the first factor that strongly affect the crop and the yields of crop. And so it is not easy is a problem this is not easy to solve, because we know that the climate change is a global, and the climate is constituted by many, many different facts. I mean, not only the temperature, not only the rain, not only the changing very quickly of environment situation, because any changing make a stress to the plants, and the plants respond. So because of course plants respond, the plants have their physiology, the plants are able to respond, and when they respond, they activate many different genes, and the complexity of all this problem is very high. I mean, the complexity is something that now can be handled by the AI, because the AI is a new instrument, is a new tool to manage many different data coming from all different disciplines. But can you see this slide? So we know that the smart agriculture is very much connected with climate. So all these part, adaptation, productivity, and mitigation, together, when they intercept each other, they give a space, something rational space, in which they make contact. So we like scientists, researchers, we have to study and to understand well this part of interaction between all these things. So also FAO was studying this big problem that is a global. So the FAO make different studies, and they make a list of suggestions. Expanding practical application of climate-smart agriculture research findings, supporting enabling policy frameworks, reinforcing territorial institutions, enhancing funding and different funding options, applying climate-smart agriculture practices in the field. This point, we think that is the real goal of this big problem. But how many tools are available and we can use to reach out the Agri-Food 4.0. Because Agri-Food 4.0 is something that we can reach after different evolution of our ability to manage agriculture. One of the first thing that we have to study is the Internet of Things. And as I say, artificial intelligence. Because artificial intelligence is able to take advantage of Internet of Things. Internet of Things means many data, big data from several points of observation. Data can be temperature, can be images from satellite camera, can be analysis from sensors by drones, can be analysis in the laboratory of physiology of plants, can be analysis of genetics data. All together, this data can be analyzed and much more than statistical, can be connected, and the connection between so many different things, like, for example, a polymorphism of one gene to the yields in the field. So putting connection these things is possible when you have a big data that are able to feed artificial intelligence, and artificial intelligence governed by human intelligence can be make the good synthesis and to find new way to overcome the big problem. So in few years, in the last 3 years, effectively, we came from agriculture machine and their monitoring system to the very difficult software, very high level software for management solutions, farm management information system, and mapping system for crops and land. There is a passage between this situation, normal agriculture machine and their monitoring system, to this one that is completely a new era, the smart agriculture era. So this approach should be able to embrace so many different technologies. The conservation of soil, minimizing soil work. The use of new germplasm lines with special genetic traits showing abiotic and biotic stress resistance. Integrated pest management, information communication technologies, and many, many other more things. Now I want to just to put the attention of this second point, new germplasm lines. Why? Because in these few words is contained many years of work of my lab. Also Kartikeyan was participating to this big project that was a long project to study the response of wheat, especially durum wheat, to the abiotic stress of water, so the scarcity of water. Here also there are a picture that show you all many contribution to this big a complex problem, and how we can solve, taking all suggestions coming from all these different discipline, different technologies, different data. So, as I say, climate change also very linked to desertification of the soil in many countries, in many continents of the world. The stress when the water is not available for plants, and especially during the development, this stress make at the level of a plant cell and also at the level of molecular cell, molecular genes, different activities. So we know that there are activating the response ABA related and also ABA independent. And in the independent way to response, there are many transcription factors that are activated by the stress. So we know, this is just a diagram explaining very easy way what's happened in a cell of plant. Oh, sorry. There is the stress, there is the signal perception, and then the signal transduction. And this signal transduction is able to activate the specific gene expression. The gene product also make the response. And the gene product can be functional proteins or regulatory proteins. Our work was much more focused on transcription factor and especially to gene belong to the DREB2 family. All these activation are able to give to the plant stress tolerant stress response. Of course, many genes are involved. So the response is a plurigenic, multigenic response. We were focused on the dehydration responsive factor, and the acronym is DRF1 gene. And we study as a case for many years this gene that we were able to isolate. We published this, I don't remember exactly, but about 2004, 2005, more or less. I don't remember exactly the year. Because this gene was a new gene. Nobody were first isolated before us. This gene is particularly interesting because is constituted by 4 exons and 3 introns. And these introns are cutted in the transcript, the long transcript, in different way, in alternative splicing. So we got 3 different transcripts, 1.1, 1.2, 1.3 transcript. And these transcripts are able to make 2 transcription factors. One is a long 393 amino acid. The other one is a long 3 and 4, 6 amino acid. And there is also a transcript that give a short or RF that is a protein without function and unable to make the function. These 2 other are instead able to activate the transcription in other gene target. Here, just very quickly, the presentation of this gene, how much is conserved, but also we found some very little differences between among different varieties, and we analyze very well in detail all the gene, all entire gene. And we study, and also we were able to identify some specific polymorphism related to the response in the field related to high yield. But this work was done and elaborated in around 20 years. So go on and we take again other information. So as I say, the meteorological data and analysis of data are so important. In fact, climate change, temperature, humidity variation affect the agriculture productivity. Thus, many predictive systems help the agriculture to defend the crop productivity and sustainability with different tools able to face every situation and minimize the damage. Climate-based solution. Many competencies work in collaboration to defend agriculture from climate change, agronomists, meteorologists, farmers, researchers, stakeholders. A climate service based on scientific knowledge has been developed to face the climate change and save the agriculture productivity system. So you can see here, it looks something that is not a solution. It's something full of information, but all these information are very important, are valid. We can use if we are able to manage and to put in relationship. In fact, one of the important suggestion is to make the precision agriculture and avoid to use some processing of the soil. One is very important new way to processing the soil is the no-tillage and the cover crop treatments. No-tillage means minimize, less is possible, the soil movement, take off the residual of the crop and so on. After many years of studies in agriculture, people understood that if you don't take off all the residue of other crop before to seed again, the soil can be improved because especially the microbiome of soil is able to use the residual and make the soil more fertile. Also, the no-tillage is able to reduce the gas emissions and increase in this way the crop yields. And indirectly, these improve food security and the resilience of the agrosystem to the climate change. Also, the cover crop treatments is very important, and that is a new agriculture method based on the use of non-productive crops between the harvest of one very important crop like wheat and the next one. So in the between, for few months, the soil can be used for non-productive crops, but some crops or some plants that are able to absorb the dangerous substance and make the cleaning of soil, to reduce in this way the soil erosion, to save water, and improve the microbiome soil community. Other important activity that we should apply are the integrated soil fertility management. This group, this name contain a group of different activities. They are applied to the agriculture with the unique scope to produce the higher yields of crop but with a lower negative effect on soil and environment. So you can understand that this is something very general, but very difficult and very important to understand. Alternate also wetting and drying. Alternate wetting drying is a method to water saving and is an eco-friendly technology. This practice is aimed to reduce the conventional irrigation system, especially those that have effect on the gas greenhouse emission, for example, for rice production. The application of this technology saves a large amount of water and soil erosion with a small loss of profit. Thus, this method also was very much studied in the last years. Another point is to reach the nitrogen use efficiency. Nitrogen use efficiency is calculated as the ratio between the amount of azotus fertilizer used by the crop and the amount of the azoto fertilizer applied to the soil. The nitrogen use efficiency is a balance and represents the value used as agro-environmental indicator of agriculture production efficiency in the conversion of the applied nitrogen in final protein content of the harvest crops. Nitrogen use efficiency is a very important component, not only at the biochemical level, plant cell physiology, and soil ecosystem, but also as a relevant cost for the farmers that has consequences on their profit. So the important note about this activity was to understand how much nitrogen remain in the soil, and the rest of this nitrogen in the soil could compromise the right physiology of the crop. So the important level of quantity of the present of nitrogen in the soil is very important, like fertilizer. And to understand that, we have need many years of study about this. Also, another point that is important, especially in these last years, is the introduction of renewable energy in agriculture. Because agriculture also, everybody knows, consume is a major activity that consume energy. And also we need to develop new method to get energy without destroy our environment. So to produce electricity, to produce biogas, but in the good way and in a renewable way, is indirectly a way to improve also our ability to manage soil, agriculture, water for agriculture. So many farm has evolved, and they put a new system to produce energy inside their company in the field, and this way they receive renewable energy without compromise the environment. So many farmers finally have successfully applied this technology, and they will have a good yield and less cost for energy. So, some conclusion. The smart agriculture is a good approach. Is also a multidisciplinary approach, but still it presents many issues to be solved. All the technologies that I have presented have a cost that often the farmers are not ready to support. Thus, right help to change the the current agriculture should be have a money, a good help from national and international institution for agriculture. Also because it is necessary to integrate research results with development. We need to make training in a comprehensive approach to people working in agriculture, including the selection and adoption of new genotypes variety best resistant adapted to the climate change. So one of the crucial problem is that the people work in agriculture. They have a necessity to improve their technologies, their abilities, their skills. And so they should be help from the national and international institution. Also, we know that in these last years, the use of bioorganic supplies, especially against pests and so on, are preferred to the traditional one. And these bioorganic supplies have the costs very high. So in the future, the trend will be to implement the climate smart agriculture approach because it is really more safe for the environment, for the soil, for the water, and for the human health. Here I just put 3 papers that I think can be very addressed to this talk. And I wish to thank you very much for your attention. And I really thank you, Kartikeyan, for this special invitation. Thank you.
Okay, ma'am. Thank you so much for your valuable and kind lecture. And it's really inspiring. So, for climatic resilience, compared to India, I don't think Italy is facing a lot of problems in case of drought or other kind of devastating factors. So, what kind of situation about the wheat, hexaploidy wheat production compared to durum wheat in Italy? Because we are the durum wheat consumer. So, you are thinking like other country, Italy, whether it is also thinking to increase the wheat production apart from durum wheat production, the barley production and other crops, minor crops, etc.
Yes, I think we have to implement and increase all these new technologies to save the crop and to make a more production. I mean, the yields sometimes are very affected for our climate change, even Italy. So this is a global problem. Of course, there are some countries, probably like yours, that is very high temperature for many long period. Italy is a little bit different, but also us, and also the problem in Italy is also the very small possibility to use the land for crop, because we are very few kilometers, very few hectares in comparison to India or other countries. So the problem is very big, and the problem could be resolved if the collaboration between all people working so different discipline working together, and also to apply the service by artificial intelligence, for example, to monitor, to make a specific day for example, for irrigation, a specific day for pharmacy, pesticide, and so on. So I think using all knowledge, all new results, all big data in the good way with these new technologies, probably we were able to overcome the problem. Also, of course, we have to continue to study genotypes, lines, the variability, the adaptation, and so on. So different.
So there must be some possibility for multi-cropping, not just monocropping, because monocropping system, it will lead to have less the genetic diversity and the other things. Italy is considered as one of the major producer of olive oil also, for example. So that can be also, India needs to think this kind of horticultural crops also very important for India to increase productivity apart from normal cereal crops. So that's my suggestion. Audience, do you have any questions? From audience?
I'm one of the audience.
Yeah, please, sir. Yeah.
Thank you, Karthik. Thank you, Deepshika. Thank you, Dr. Patrizia, for such vast knowledge that you imposed on us by keeping the view of climate resilient agriculture. Alternatively, you called it climate smart agriculture. Very nice, madam. And wherein you have shown your command to deliberate the things by deploying the IBM, ICT in terms of holistic approach, that what do we can do to alleviate all the stresses in the soil, which may be amicable for the crop fertility and henceforth soil fertility as well. Madam, another interesting point that I wanted to unravel the perplexity of nitrogen use efficiency that you defined somewhere. So that is a one more important parameter. Along with this, I like towards renewable energy agriculture, REA, wherein you disseminate the knowledge about the agrivoltaic deployment. Lastly, you defined the use of biofertilizer and biopesticides in toto. By deploying these two, how do they show the syntrophy or the co-metabolism in the soil, which may helps in the enhancement of the soil fertility? So, madam, I would like to see that if we see the temperate and the tropical region of India, what climatic conditions prevails in Italy, either tropical or the temperate or in between? Would you like to provide some glance on it, please?
Okay, Professor Chudari, thank you for your question and your introduction to the question. It's okay if I understood all your point of view. I think Italy, of course, is very different climate from India. So we are not tropical, we are Mediterranean area, and the general climate was temperate. But Italy especially has, because it's very long country, is like a boot, the shape.
Sure.
We are about 2,000 kilometers in length.
Right.
And it's very small, but we have very strict. But we have also a very important mountain, the Alps on the top and the Apennino in the center. This big mountain give changing the climate. I mean, for example, Rome, where I live, in the region is Lazio, we are very protected from mountain of Appennino, and in front we have the Mediterranean Sea that give something the climate sweet, you know, it's not too cold, it's not too hot.
Okay.
Now, in this more or less 20 years recently, the summer for us is very high, more than sometimes more than 40 degrees. When I was young, never was so hot. Could be 30 degrees in the summer. And also we had always a very nice wind. So sometimes the air was fresh, or even in the summer you have good temperature but fresh air. In the 20 last years, this wind is not present. The air is very heavy because it's very wet. Sometimes we have 80%, 90% wet, 70%. It's too much. People suffer very much the climate. So how we suffer, even in the field, plants are suffering. And for example, in Italy, in Lazio, the triticale will be harvested before in July. Now they harvest in May, June, because it's too hot. Okay, so even in country different, we are going to see a very strong negative effect of the climate change. So agriculture must solve the problem. And we can do it. We are able to do all together, scientists, people expert of agriculture in the field, all technologies that help agriculture. So all together, it's possible with a smart agriculture to solve the problem.
Right, right. To reduce the greenhouse gases as well, as you discussed, right? Right.
Yes, yes.
Okay. Thank you, doctor. Thank you.
Thank you to you and to all your colleagues and students. Very, very, very kind from you, this invitation. Thank you very much.
Reciprocally, reciprocal sentiments to you as well. Thank you. Thank you, madam.
Thank you.
Dr. Deepshikha? Dr. Deepshikha, please.
Yes, sir. Yes, sir.
Please.
Dr. Karthikeyan, please proceed.
हाँ कर दीजिए कल हाँ कल के लिए मिलाएंगे फिर
Dear participants, am I audible?
Yes, you are audible.
Am I audible?
Yeah.
Sir, am I audible?
Yes, yes.
Okay.
Yes.
So dear participants, thank you so much. Today we had only one session in the afternoon, and thank you for your patient listening, and thank you all the speakers that we had today. Patricia ma'am, thank you so much for joining us and for wonderful insights that you have shared with all the participants. Thanks a lot. And with this, we will end up today's session, and we will meet tomorrow morning for the next fresh, most awaited sessions. Thank you so much. With this, we
Thank
will
you.
end the end day one of the presentation.
Right.
Thank you
Thank
to
you.
all. Thank you.
Thank you.
Bye.
Thank you.
Bye.
Bye.
Thank you. Thank you, ma'am.